Metabolism. That's our subject today. Let me share my screen because I want to show you something before I start actually getting into the notes. So let me just shrink this down a little bit. Let me get into course content, and then I will show you where this document is found. Okay, and let me just see if I can. It's me a minute. Okay, here we go. All right, so I'm in exam 2 material. Pardon me. And we talked about the digestive system, the digestive systems chart. Well, I'll briefly go over that with you. This is how I organize the enzymes of digestion. Of course, it's sideways, but for each column, we have carbohydrates, proteins, lipids, and nucleic acids, identifying the building blocks, the intermediate, the macromolecules. And in doing so, when you do it this way, you can, with arrows, beginning at a polysaccharide, ending with a disaccharide to a monosaccharide. I mean, this is what digestion is, starting with large molecules and ending with smaller ones. So when I draw it out this way, and the arrow always points to the product. So if I'm starting with a polysaccharide and with a disaccharide, I'm going to be pointing to those. To the right and left, I have color-coded the enzymes and where they're produced. So facilitating or chemically and enzymatically, hydrolytically breaking down the polysaccharides to your disaccharides is salivary amylase produced in the mouth. And we said pancreatic amylase from the pancreas does the same thing. It's just that, when you swallow the food, the enzymes become denatured by the stomach acids. Okay, then taking from a disaccharide to a monosaccharide, we have maltose, sucrose, lactose. Then maltose is broken down by maltase, sucrase to sucrose, lactase, acts on lactose. And those are the respective individual building blocks of each of those. So here, between the disaccharide to the monosaccharide, we have lactase, sucrase, maltase, and producing the different combinations of two monosaccharides for those particular disaccharides. I don't generally ask you questions about that. Well, I guess I could. I could ask you that, in lecture. Sometimes that's more of a lab question because I use the models and things, but yeah, I guess I could. I could ask, what would be the final products of digestion of maltose? That would be 2 glucoses, or what's the final product of digestion of lactose by lactase, a glucose and a galactose. I guess that's possible. Polypeptides, we start with proteins. We have pepsin, trypsin, and chymotrypsin. And then acting on the dipeptide, we have carboxypeptidase from the pancreas also, and dipeptidase from the small intestine. Finally, bring that dipeptide down to its amino acids, and so forth. So, that might help you. I think whenever I organize and color code, that works for me, but that's entirely up to you. Okay, but let me go back to the exam 2 material, which is really relevant for this lecture, and that is I have digestion metabolism review questions, and the answers here. So we have I have review questions that you can use to accompany the notes, so that as you read the question, they kind of parallel areas of the notes. Beginning at the beginning of the notes and going all the way to the end of the PowerPoint, there are different, questions asked on the content. So that's what I would do. Let me get back into my notes. And we'll go here to metabolism. And I will also say that given those review questions, that I generally limit my questions to those questions, meaning if you answer the questions, the review questions, then you will be able to answer really any question I have on the quiz or the exam. Okay, so let me just put this in slideshow mode and we'll get crack-a-lackin'. Okay, as an overview, anabolism are the different reactions that combine monomers to form complex molecules by a different dehydration synthesis. Remember, dehydration synthesis is when we remove the equivalent of one water molecule, create a bond between two molecules, and create a larger molecule. And we store energy in the bonds that we create. That means that it's an endergonic reaction, which means we're going to put energy in and store it in the bonds. Now we can release it later in a catabolic reaction, and otherwise referred to as an exergonic reaction. So catabolism, chemical reactions that are chemical reactions that break down complex molecules into smaller ones. And we do that by hydrolysis. Add water and break apart. They are exergonic reactions because we release stored energy. Most energy is lost as heat, and the rest of the energy is transferred to ATP. We take an inorganic phosphate, we put it together with an adenosine diphosphate, to create adenosine triphosphate. Right? ATP is used for anabolic reactions or for cell work, muscle contraction, and active transport. So we need ATP. It's the energy currency of the cell. ATP is going to pay for all these different active processes. So we have to make it in-house, so to speak, in our mitochondria. We can take in different food sources, carbohydrates, proteins, and fats, and use them to create energy, to create this ATP. Now, they come into these different reactions at different points, because we're going to talk about these four, phases of cellular respiration. But depending on whether we're trying to burn proteins or if we're burning carbohydrates or fats, and I will say that carbohydrates, glucose is the preferred molecule, but we can clearly burn fats because that's what one of the goals is to, exercising for long periods of time. If you're trying to lose weight, you want to be in aerobic respiration and go from burning carbohydrates, to burning fats. And then, we also can burn proteins. But they just come in, at certain phases in glycolysis or acetyl-CoA, or in the Krebs cycle. Most fats will just join, here. The glycerol portion can enter into the glycolysis, the fatty acids, into the formation of acetyl-CoA. So there's different phases in which these molecules can use, but glucose and other sugars are used from the beginning of glycolysis and is the preferred molecule. Okay, so carbohydrate catabolism is in cellular respiration. All carbs are broken down, ultimately to the monosaccharides, to their building blocks. And is the common or is the same chemical formula, C6H12O6. Depending on how that molecule is arranged, it may be a glucose, a fructose, or a galactose, but they all have the same chemical formula. And glucose is the preferred source for synthesizing ATP. Insulin is needed for glucose uptake into the cell, as you know. Okay, so ATP synthesis. There are two mechanisms in which we can make ATP, in which we do when we look at all these different reactions. And, mind you, these reactions and all this information can be a little overwhelming. But if you accompany these notes and maybe listening to this and have the review questions, you just fill out the review questions as you go. And that's essentially organizes all this into the types of questions I might ask you on a quiz or on an exam. Right, so there's two ways to make ATP. You can do it through what's called substrate phosphorylation or oxidative phosphorylation. And substrate phosphorylation means that we're going to, high energy phosphate groups are going to be directly transferred from a phosphorylated substrate to ADP. So we're going to take an inorganic phosphate, we're going to put it to an ADP, and this happens twice. Twice in glycolysis and once in Krebs cycle, where we're producing ATP by having an ADP there and a phosphate group, and we put them together. All right. Oxidative phosphorylation, is a more complex process, but produces the most ATP. right? This uses what's called the chemiosmotic process. It couples the movement of substances across a membrane, to chemical reactions. So what it's going to do at the end of all these four, not all these, but these four different phases of cellular respiration is that the last phase produces the most ATP, and you'll see how this method of hydrogen ions flowing through what I call, it looks like a turnstile, will create the energy, to form the ATP. And in doing so, it creates, 30 to 32 molecules of it, the most abundant. All right, so we have, and I'll identify the different stages where we're producing ATP through substrate level phosphorylation or oxidative phosphorylation. This is required to know for microbiology. I just went blank. Okay. So we're going to start with glucose catabolism, breaking down glucose. Glucose catabolism is what we refer to as cellular respiration. We're going to have glucose and we're going to have oxygen. It's a 4-step reaction sequence. We have glycolysis, which occurs in the cytosol. We have what's called formation of acetyl-CoA, which is essentially a link reaction. When I took organic chemistry way back, we never considered this, or this wasn't treated as really a separate reaction, but in textbooks now, they do. It's called a link reaction. Then we have the Krebs cycle, which is also referred to as a citric acid cycle, and the electron transport chain. Okay, so as an overview, so to speak, here we have the mitochondria. Remember, in the models in lab, they were like the abundant red jelly beans on those different cell models. We have the outer membrane to it, and then we have what's called the cristae, which are these folds within it. Glycolysis is going to occur outside the mitochondria in the cytoplasm. And then depending on oxygen availability, we will have this linker reaction that goes into the mitochondria, and it's going to give rise to the citric acid cycle, and then ultimately the electron transport chain. The citric acid cycle produces a little bit of ATP. The glycolysis also a little ATP, but over to the right, the big ATP sign there, it's trying to indicate to you visually that the majority of ATP is going to be produced in that last phase. Glycolysis occurs in the cytosol, breaks down each glucose molecule into 2 molecules of pyruvate or pyruvic acid. The pyruvic acid enters into the mitochondrial matrix where the citric acid cycle oxidizes it to carbon dioxide. During glycolysis in the citric acid cycle, substrate level phosphorylation forms small amounts of ATP. That's all it's saying. But in the last step here, energy-rich electrons picked up by coenzymes are transferred to the electron transport chain. It's going to pass through this little turnstile and use the concentration gradient flowing from where it's in high concentration to low to generate the most ATP. So that method is different. The first 2 sources of ATP in glycolysis and the citric acid cycle is via substrate phosphorylation. And then the last is oxidative phosphorylation. Don't worry really about all this writing down here in the bottom in black. I will tell you what you need to know. Okay, starting off with glycolysis, we're going to take a glucose molecule, which is a 6-carbon chain, and essentially split it into 2 3-carbon chains. It's going to go through a series, of complicated reactions to rearrange it and so forth. But ultimately, we start with 1 glucose molecule, a 6-carbon chain. We end with 2 3-carbon chains. And then from here on out, we're going to follow these 2 pyruvic chains. So everything that's produced, we kind of have to double because we have 2 3-carbon molecules instead of just 1 6-carbon molecule. Okay, so this first reaction does not require any oxygen, so it's considered anaerobic. Eventually, we're going to need the oxygen. It occurs in the cytosol of all living things. NADH transports high energy electrons to the electron transport the cycle. in the mitochondria where we will reap lots of ATP, electron transport chain. I don't know why I can't say that. All right, so NADH is going to be kind of a vitamin like FADH that is going to transport hydrogens. That's all it's meant to do. It's meant to bind with a hydrogen here and drop it off over there. And then it goes back again, grabs a hydrogen and transports it to the last phase in the electron transport chain. FADH is going to do the same thing, or FADH2. Pyruvic acid conversion depends on oxygen. So we start with a 6-carbon, we end with 2 3-carbon chains. Now we have to go through a, this intermediate kind of step where we are oxygen dependent. So if we want to convert that at all, we have to have oxygen, and then we enter into the mitochondria. If we don't have oxygen, like when you're exercising and you're exercising really strenuously and you start to go into anaerobic respiration, it's because you're building up lactic acid. So when we don't have oxygen and we're breaking down this glucose, it's going to be converted to lactic acid. Now that can be reconverted, to pyruvic acid in the liver, but it's going to take a little bit, for that to happen. Aerobic means if we have oxygen, it's going to be aerobic, and it diffuses in the mitochondria for the next sequence of reactions. The ATP that is formed within glycolysis is via substrate level phosphorylation. So the method is substrate level phosphorylation. So what are the products of glycolysis? We start with one glucose molecule, right, a 6-carbon chain, and we end with these things. So know what you begin with, what you end with, and that substrate level phosphorylation occurs here. Because we have 2, and we're going to end up with 2 of these pyruvates. We have 2 ATP that are produced, 2 pyruvate to 2 3-carbon chains, and 2 NADH. So the NADH is transporting that hydrogen. We now have a 3-carbon chain, 2 of those molecules, and along the pathway, we produce 2 ATP. Well, let's put it this way: we netted 2 ATP. We actually produced 4, but it cost 2, so we are ahead 2 ATP molecules. All right, so you can see here, where glycolysis is occurring out in the cytoplasm, and glucose oxidation and ATP formation. They're just showing you here that at a certain stage, right, we are producing and peeling off 4 ATP here. Now, I will tell you that along the way, we had to add 2 ATP. So we had to invest 2 ATP to get 4 out. Excuse me. And so we have actually, our net ATP is 2 molecules for glycolysis. Okay, now what we're going to do is we're going to take those 2 3-carbon chains and we're going to form or produce what's called acetyl-CoA. All right, so we're going to remove the equivalent of 1 carbon dioxide from each of these and let it diffuse into the blood. Because you keep wondering, like, well, where does all this carbon dioxide come from that we have to exhale away? It comes from, the aerobic respiration, cellular respiration, producing ATP from glucose. So the carbon dioxide is going to diffuse into the blood. And we're going to produce some more NADH, 1 for each of these 2 molecules that's going to have to be converted. Hydrogens and electrons attached to the NAD for, again, transport it and take it down to the electron transport chain. Acetic acid is attached to a coenzyme A, or what's called a CoA for transport, and forms acetyl-CoA. So we have this acetic acid, at the end of rearranging this pyruvic acid, or the pyruvate molecule, because we took a carbon dioxide off. Now we're down to a 2-carbon chain, essentially. And that's what acetic acid is. If we put this coenzyme A on it, it's acetyl-CoA. All right, so acetyl-CoA now can enter into the Krebs cycle, or the citric acid cycle. So we started with 2 3-carbon chains of 2-pyruvic acid. What did we produce? 2 carbon dioxides, 1 carbon dioxide from one of those pyruvates, 1 from the other. We also produce 1 NADH from each of those 2 that entered this phase. Right? So 2 NADHs, and now we end up with 2 acetyl-CoAs. Right, then we enter into the Krebs cycle. The Krebs cycle means we're going to go around in a circle. So our 2 acetyl-CoA are going to now enter into the Krebs cycle, and it's going to join what's called like this 4-carbon chain to restore the 6-carbon chain that we kind of started out with. But now we're going to have 2 of them. One crank around this wheel, this cycle for each of these acetyl-CoA that's going to enter. And it goes around and around and around. Because 2 acetyl-CoA molecules are produced from each glucose molecule, 2 cycles are required per glucose molecule. Called a cycle because the beginning molecule, oxaloacetate, is regenerated by the end of the cycle. And so as a consequence of the 2 acetyl-CoA entering into the Krebs cycle and 2 cranks around, 1 for each acetyl-CoA, we produce 4 carbon dioxides, 2 from each of the acetyl-CoAs, 2 ATP via substrate phosphorylation, just like we did in glycolysis, 1 ATP per acetyl-CoA. We have 6 NADH transporting hydrogens that we produced, and we're going to transport it down to the electron transport chain, and a new molecule, 2 FADH2. Right? They're going to also bind to these hydrogens and transport them down to the last phase. All right, so as a total, what enters Krebs cycle? 2 acetyl-CoA. What's produced? 4 CO2, 2 ATP, 6 NADH, 3 NADH from each acetyl-CoA, and 1 FADH2 from each acetyl-CoA, producing a total of 2 FADH2. In this cycle, we had these intermediate molecules, how would you say that? Excuse me. And the mnemonic I found that helps me keep this in order is, can I keep selling sex for money, officer? Now, you still have to know what each stands for, but it keeps, each letter, but it keeps them in sequence so you don't get them confused. So as a consequence of acetyl-CoA binding to oxaloacetate, which is actually on the bottom, it forms a citrate molecule. Then it gets rearranged to form an isocitrate and rearranged for the alpha-ketoglutarate, and then a succinyl-CoA, a succinate, a fumarate, a malate, and then you end with oxaloacetate at the end. So I've just found that helps to keep them in sequence. You still have to know what the I, the C, and the S's, the 2 S's stand for, but if you can practice this will help keep you on track. Okay, so this is a simplified version of the Krebs cycle. This is up on the top. We have up on the top in the cytosol, in the blue, all right, this is where glycolysis occurred. We started with glucose, a 6-carbon chain. We ended up with 2 pyruvates, 2 3-carbon chains. We have oxygen, so it goes into the green area in the mitochondria. And when we start with the pyruvate, we are going to end with acetyl-CoA. Right? We're going to take off a carbon dioxide, which is the black arrow to the left. Where we have production of NADH, which is the black arrow on the right. And we add a CoA in the blue ball there, and that forms acetyl-CoA. All right, now we have 2 of those entering into the cycle here in the green arrows. So that merges with, to the left, oxaloacetate or oxaloacetic acid. It's a 4-carbon chain here, 4 black balls, and it merges with the acetyl-CoA to form citric acid. Right? So the 2 green arrows merge, form a citric acid. That's your initial reaction. And it produces, here the CoA actually does produce, but the CoA comes off. Okay. So now we have from citric acid, it just gets rearranged to isocitric acid or isocitrate. Now, this is where we first start producing our carbon dioxide. So from isocitrate to alpha-ketoglutarate or alpha-ketogluteric acid, we remove carbon dioxide and we produce an NADH. Then we have alpha-ketoglutarate to succinyl-CoA. Happens again, remove a carbon dioxide, produce an NADH. From succinyl-CoA to succinic acid, we are doing a lot of rearranging and producing our ATP there. From succinate to fumarate or fumaric acid, that's where we produce our FADH2. From fumarate or fumaric acid to malic acid, we're just rearranging. And from malic acid to oxaloacetate, we're producing another NADH. All right, so you can see how now we restore our 4-carbon chain here, and we're ready to merge with another acetyl-CoA. Sorry about that. I mute my phone, and it only mutes the text and not the telephone portion. And so for each molecule of acetyl-CoA, which we have 2, it's going to be 2 turns around this cycle. Now, I'm not going to ask you where, between which phases do I produce a carbon dioxide, or, between isocitrate to alpha-ketoglutarate, what is produced. I just want you to see how this works. I would try to memorize those intermediate molecules in sequence, because what gets added and taken away and rearranged and so forth is really the subject for more organic chemistry and so forth. But it's good to know those intermittent phases, because in microbiology, you're going to talk about, hey, when I'm trying to break down burned fats or proteins, in this cycle, they're going to come in at this molecule or that molecule, and so it'll be helpful that way. But I'm going to ask you questions about what you start with, what you end with, okay? Or maybe what type of, ATP production was there or was present, that formed that ATP, substrate phosphorylation. Okay, so, all those NADHs and FADH2s that you produced, well, this is where they become relevant. You produce those just so you can use them at the end. Right. So those NADH and FADHs are coenzymes that transport electrons and hydrogens from each of the previous, reactions. As a consequence, you produce, and it's not, I will say that these are the averages. It's not always exactly the same all the time, but it's pretty much the same. If we're burning glucose, then we'll have produced about 10 NADH, 2 FADH2s, transporting hydrogens and their electrons to this electron transport chain. And these electrons enter the chain through a series of enzyme-regulated steps, and they're going to get shunted across this inner membrane of the mitochondria. Oxygen is required for this step as well. So this is another oxygen-dependent point of cellular respiration. ATP is synthesized as electrons flow along the chain through this, it's called a chemiosmosis, and passes through this little turnstile, it looks like to me. And at the end of the chain, oxygen picks up the electrons. It's called the final electron acceptor. Remember, they're electron because hydrogen has an atomic number of 1, which means it has 1 proton, and it had 1 electron, but it's an ion. And so all it is a traveling proton, positive charge. Okay. So hydrogen and depleted electrons bind with oxygen to form water, and that's going to be one of the byproducts of this as well. When we produce ATP in this manner, it's called oxidative phosphorylation. And the total is about 30 to 32 ATP and water. All right, so let's just look at this. So what we're saying here is here in the upper left-hand corner, they're showing you the mitochondria. You have the outer membrane, and then you see you have what's called the cristae. Looks like waves of tissue in there. And then in the upper right-hand corner, it goes, okay, glycolysis is occurring outside in the cytoplasm. Excuse me. So it's not oxygen dependent, but if you have oxygen, then you're going to produce that acetyl-CoA, and acetyl-CoA is then going to enter in the Krebs cycle, and now we're inside the mitochondria. All the NADHs and FADHs that we produced in glycolysis, formation of acetyl-CoA, and Krebs cycle all get used here. Right? We produce a little bit of ATP, a little bit, but at the end is where we produce the abundant amount of ATP. So now let's go into where these different enzymes are. It has a Roman numeral of 1, 3, and 4. And so not that you really, I'm not going to ask you about this, but the NADHs kind of get torn apart and their hydrogens get taken from them. And it gets pumped from one side, because you can see that phospholipid bilayer here. That's the composition of this inner undulating membrane here called the cristae. And it's going to be pumped to the other side. So now you're in between the outer membrane of this mitochondria and the inner membrane, this little gap area kind of here. And then when the FADHs enter, same thing. You take the hydrogens and you pump them out. So they're in between, these hydrogens are in between these two membranes. Toward the end, over to the right-hand side, you see this funny little, molecule here. To me, it looks like a turnstile, like you're going through the turnstile to get on the subway in New York. The hydrogens are going to fall through here, and that will create the energy to bind together an inorganic phosphate to ADP and produce ATP. It's also going to produce a lot of water. So this method of pumping hydrogens out and letting them fall through this turnstile to create the energy to bind, the inorganic phosphate to the adenosine diphosphate, that's a different method called oxidative phosphorylation versus substrate phosphorylation that occurred previously. And so you can read on the bottom there, there's more detail to this. I'm not going to ask you, excuse me, about that detail. But I will ask you, what do you start with? What do you end with? Okay. This gives you another picture. Like, it just blows up that little turnstile a little bit more. It's like a little rotor in there. And as it passes through, again, it will be the energy that creates the ATP. All right, so, just kind of by going over this, we have we net, as I said before, 2 ATP in glycolysis. You net 2 ATP by substrate phosphorylation, and then you have about 20, and different books will give you different amounts. Some will say 30 to 32, some will be 28. So I would say 30 to 36 ATP produced in total is not uncommon. Somewhere around 30 to 32, 34 or 36. As you can see over here, this is one of those little details. NADH produces about 2.5 ATP per molecule. FADH about 1.5 ATP per molecule. So even though these are both transporting hydrogens, they end up generally generating different amounts of ATP too. So in any event, if you know those basic numbers here, you should be fine because it is multiple choice. Okay, so let's talk a little bit about lipid transport, because we're going to talk about these other remaining molecules, with some information about the lipids and proteins and their metabolism, and then we're done. All right, so lipid transport. Remember, it's non-polar covalently bonded, so it's not soluble in water and combines with proteins to allow transport called lipoproteins or lipoproteins. If we didn't have those lipoproteins, they wouldn't be soluble at all. All right, so depending on the ratio of fat to protein will determine the density. Right, these are the different types of lipoproteins. Right? And the more protein you have, the heavier it is, the more dense it is. Let's put it that way. So we have chylomicrons, excuse me, pardon me, VLDLs, which are very low density lipoproteins, low density lipoproteins, and high density lipoproteins. chylomicrons, as these fats are being transported through the columnar cells, that simple layer of columnar cells lining the small intestine, right? That's where we produce these chylomicrons in the smooth endoplasmic reticulum. And I swear I had that in here at some point. Right. So yeah, in the SCR of columnar cells, they will be released on, exocytotically released, and they will enter into the lacteals, where then they ultimately get transported to the liver. Remember, they're going to go through the lymphatic system to the blood and pass through there. Very low density lipoproteins, are formed by hepatocytes. So now these molecules are formed by cells of the liver. They transport triglycerides formed in the liver to adipose tissue. Remember, once you're reaching adulthood, the number of adipocytes doesn't really change. They either store more or store less. You either blow them up like a balloon or let them deflate a little bit. And unless you get liposuction or something, or some other method of killing off the adipocytes, you're going to have the same number of them. And so they will store triglycerides. Low density lipoproteins carry about 75% of the body's cholesterol and is transported to all body cells. At high levels, they can deposit cholesterol in smooth muscles of arteries and things. So, you want to keep your low density lipoproteins low and your high density lipoproteins, percentage wise high. Right? Because they have a tendency to deposit cholesterol in smooth muscles, especially those, that are the smooth muscles of arteries. Okay, high density lipoproteins, or HDLs, remove excess cholesterol from cells and from blood, transport to the liver for elimination. Right, so HDLs are working for you to help get rid of excess cholesterol. Whereas low density lipoproteins is depositing cholesterol, or has the capability of depositing cholesterol to the smooth muscles, but transported to all body cells. So how do we break down triglycerides? Well, that was the job of lipase, right? We said lipase from the pancreas will break it down. Your triglycerides, two building blocks, one glycerol and three fatty acids. Technically it's one glycerol and one fatty acid, and then the two fatty acid tails, but we're not going to pick. Fatty acid chains can be further broken down, though, and you do that via what's called beta oxidation. So the reason why I want to talk about this is that you do a lot of this beta oxidation, breaking down these fatty acids when you are not burning glucose as your primary molecule. And for diabetics that aren't being, like diabetes mellitus patients, I guess I should say, are not burning glucose adequately because you are either insulin resistant, maybe you do have a change in insulin production, or you were born, or you inherited that in diabetes 1, diabetes mellitus 1, where you have an autoimmune disorder. So you're not burning, or you don't have the insulin available to stimulate your cells to take up the glucose and burn that for ATP. It will use other fats and proteins for that. Now, you won't necessarily, if you're managing your diabetes, appropriately, you won't be burning a lot of these fats, but if you're not, then you can have what's called ketoacidosis. Okay, so beta oxidation, or fatty acid catabolism, is used to make acetyl-CoA, which enters into the Krebs cycle. So it has a function. This is how we're going to break down some fats and use some of it, because it can act like acetyl-CoA, or is used to make acetyl-CoA, and it'll enter into that, the Krebs cycle right at the top where it binds with oxaloacetate. It also produces ketone bodies, so two acetyl groups combined. Right? If fat levels are too high or carbs too low, right, beta oxidation rates increase and high levels of ketone bodies are produced. That can cause what's called ketosis. Ketosis causes blood pH levels to drop and can lead to acidosis, or ketoacidosis, abnormally low blood pH. Okay, so these two ketone bodies, these will, when you're burning these fats, you're ultimately going to produce these acids, these keto acids. And the more acids you produce, we said that acids break apart and liberate hydrogen ions. increase your hydrogen ion concentration. It does what to pH? It decreases your pH. So it can lead to what's called ketoacidosis, a type of metabolic acidosis. It's not good. That's not, you don't want that. All right. Now, even though, let me just go back for a minute and just say, these keto foods, where you're doing like you're purposely trying to burn more fats and put yourself into ketosis, that ketotic state, isn't extraordinary. For diabetes mellitus patients, they don't have enough insulin, they're burning these fats. They really have no choice because they have to produce, energy. But they do so at a cost. Now, when you're on like a keto diet, you're trying to shift to burn more fat than glucose, but you do produce insulin. I mean, you are taking in carbohydrates every day, just not as much as the average person. You really want to restrict it so that you're burning the fats, and that hopefully, the point is that you lose some fat and you lose weight and you become, at least have less stored fat. Let's put it that way. Not necessarily healthier, but less stored fat. So that you can check, by checking your urine, because urine represents everything that's overflowing, and getting a lot of the leftover metabolic products, or the products of metabolism, occurring in the body. Those ketones will overflow, and so you can check your urine and see if you're in ketosis. And it's going to be color coded, just like any urine strip, where it'll give you an indication about how many molecules per microliter or whatever, micrograms, whatever the unit of measurement is. You may be mildly ketotic or strongly ketotic, but it does indicate that you're burning fats. Okay, lipid anabolism, or creating fats, that we can start with glucose, end with glycerol and triglycerides. So what do we mean by that? It means that even if you're not eating fat, even if you have a very high, let's say you have a high carb and protein diet and you ate very, very few fats, if you still take in more calories than you're going to burn that day, it gets converted to fat, right, in the liver. I mean, even though it might be a better source of calories in excess on a daily basis than you burn off gets stored, as fat. At first, the glucose or let's say, like carbohydrates that you eat will be stored as glycogen, but if you continue to take in more calories that day, it gets converted to fat. That is stimulated by insulin, and occurs in liver and adipose cells. Right, protein breakdown or protein catabolism. Right, well, we just looked at that in digestion, right? We said proteins, get broken down to dipeptides and ultimately to the building blocks of amino acids. If there was a series of enzymes, right? We have pepsin in the stomach, we have trypsin, chymotrypsin, we have carboxypeptidase and dipeptidase. These amino acids, if we're going to burn, because we can burn protein, clearly for energy, they can enter either into glycolysis, the Krebs cycle, or as acetyl-CoA. Now, that must be deaminated, which means that, remember when, I don't know if you had me for AMP1, but when we were looking at the models for amino acids, we said that you knew one amino acid, one little unit had one blue ball associated with it, because we said proteins had blue balls. So if you saw one blue ball and this little double bond, then you knew you had one unit, which would be an amino acid. If you see two blue balls with these double bonds between these other carbons and whatnot, then you had a dipeptide, and you saw a string of blue balls, you had a polypeptide. Okay. Well, that blue ball is the nitrogen, and that needs to be removed. So deamination means you lose that NH2 in the liver. And that's its job. So it takes the NH2, it can bind it with another hydrogen and form ammonia. And then ammonia can be transported to the kidneys, where we can eliminate it as urea. So protein catabolism requires deamination. We need to cleave off that, once we're going to start burning this amino acid, is we need to get that blue ball off, essentially, the nitrogen off and excrete it in urine. We create proteins by taking amino acids and binding them together for dipeptides and larger proteins, polypeptides, and we do that a lot in the liver to make things like albumin. Remember that the ribosomes, which perform amino acid assembly in cells, they can do that on their own there. The liver will use proteins to form your plasma proteins like albumin, fibrinogen, and different antibodies. Metabolic regulation. So now we're getting to the last one, folks. I want you to know the difference between your absorptive states and your post-absorptive states. The absorptive state, is when all these nutrients are entering into your bloodstream after a meal. And this is when your glucose levels are generally the highest within a few hours after eating. So within, 4 hours of ingestion. And it kind of depends on how much you ate too. Right? The dominant hormone is insulin. Why? Because glucose levels will be going up, and that stimulates the beta cells to release insulin. Once cell stores glucose, extra glucose is converted into fat, and that's lipogenesis. Overall effect will be to decrease blood glucose levels. Right? You're going to store glucose in the liver as glycogen. You may start producing fat. You let body cells take up whatever available glucose there is, give them first dibs, and then what's left over after storage and things is going to be converted to fat. Right. The post-absorptive stage is after this 4 hours, 5 hours or so. After a few hours, after a meal, when absorption is complete, or it's called your fasting state. So now that you've taken up the glucose and you've stored it all over, is that okay? Your glucose levels are normal, back to a normal range. But and you're actively doing things for a few more hours. Well, now the cells are using up all that glucose, and your glucose levels are now dropping below normal, which is going to happen. Glucose levels are very volatile all day long. So what do we want to do? We want to increase our glucose levels again, right? So we're going to secrete glucagon and maybe even cortisol. Glucagon is going to be the primary. Cortisol helps increase glucose levels as well. Overall effect is to elevate blood glucose back to a normal level. So the absorptive stage, the first 4 hours, insulin, and the overall effect is to decrease glucose levels. The post-absorptive state, is after about 4 hours or so. Now your glucose levels are too low. Dominant hormone is going to be glucagon, and we use some cortisol, but you don't have to have cortisol secreted. You can use glucagon. And the overall effect is to increase blood glucose levels, and maintain appropriate level. All right, and that's it for metabolism.